Navigating the genetic maze, understanding our blueprint for life. Welcome to the fascinating world of genetics, where every twist and turn can lead to new discoveries about who we are. Imagine navigating a complex maze, with each path representing different aspects of our genetic material. Let's embark on this journey together. At the heart of our maze is the genetic material, the blueprint for life itself. This blueprint is called DNA, which stands for deoxyribonucleic acid. DNA contains the instructions that guide how we grow, develop, and function, like a map guiding us through a maze. Let's start with cells. Cells are the basic building blocks of all living organisms, and they're responsible for carrying out all the functions that keep us alive. Every cell in your body contains important resources needed for survival. It's within these cells that your genetic information is stored. Imagine your body as a large, intricate maze. Each part of the maze has a specific role, and the goal is to navigate it smoothly. The cells are like the farms where the maze is located. These farms house everything your body needs to keep things running efficiently. Inside each of your cells, your genetic information is organized into structures called chromosomes. Chromosomes are like large containers that organize and protect the genetic material in humans. There are 46 chromosomes grouped into 23 pairs, one set from your mother and one from your father. These chromosomes divide and organize the genetic information to ensure that everything works properly in the body. So within the maze, you may have distinct regions or zones. We can think of these as the chromosomes. Each region holds part of the maze information organized and protected to keep things running smoothly. These regions help to divide up the maze into manageable sections, keeping everything in order, just like your chromosomes. Within each chromosome are DNA, long, coiled molecules that carry the instructions for building and maintaining the body. DNA contains the detailed blueprint of your body, encoding all the information that tells your cells how to function, grow, and divide. Within each region of the maze, or the chromosome, you have maps, which are the DNA. Each map provides detailed instructions for how to navigate the maze and keep it up and running properly. It's the blueprint that guides every process, making sure everything happens in the right order. And within the DNA itself are genes. So what are genes and what do they do? Genes are sections of DNA that contain specific instructions for creating proteins, which perform many essential roles in your body. These proteins are responsible for everything from building muscles to protecting you from infections. Each gene carries the instructions for a particular trait or function, like determining your eye color or how your body builds and maintains its structure. So finally, within the maze map of your DNA, you have individual pathways, which are the genes. Just as a maze might have hundreds of different pathways, your DNA has thousands of different genes. Each path in the maze leads to a specific destination guiding you through the maze, maybe helping to clear obstacles, providing secret passages, or even adapting to new challenges. Some pathways might be predetermined, and some might be triggered by environmental factors or obstacles. But in essence, all of these individual pathways lead you to different destinations and have different functions. Interestingly, everyone essentially has the same number of genes, over 20,000. Each person has a similar blueprint for their genetic maze, but differences arise not from the number of genes, but from variations within those genes. These variations are like detours or blockages in a path. Sometimes a change in a gene can stop it from working properly. When this happens, it can affect how proteins functions, leading to health issues or different traits. So we discussed how important genes are because they carry the instructions for creating proteins. But what are proteins? Proteins in our body play crucial roles, like helping to transport our blood through our bodies to protecting us from illness. They're not the same as the protein we get from food. Instead, these proteins are made by our cells to keep us healthy. So if a gene that codes for a protein has a change in it, causing it not to work properly, then the protein is no longer working as intended, and this may lead to adverse physical symptoms. Using an example, in the case of Duchenne muscular dystrophy, or DMD, proteins play crucial roles in muscle function and development. But the condition arises due to a mutation in one specific protein, dystrophin. So, let's say the DMD gene that is supposed to create the protein dystrophin, which is important for muscle function and structure, has a variation. It does not produce the protein effectively, resulting in muscle fiber damage, inflammation, and the eventual breakdown of muscle tissue. Understanding that everyone has a similar number of genes helps highlight that the differences in our health and traits come from these variations. Some pathways might work perfectly, while others might need rerouting due to genetic changes. This knowledge can help demystify why certain conditions occur and reinforce the idea that our individual journeys through the maze of life can be quite different, even if the basic layout remains the same. Now that we know the role of our genes and proteins, let's look more at the genetic changes or variations. You may have noticed that we've used the words mutation and variant in this video. To clarify their meanings, generally, a mutation refers to a permanent change in the DNA sequence, often associated with disease or abnormal function, while a variant is a broader term that refers to any difference in DNA sequence, which may or may not have a harmful effect. Like different paths in a maze, genetic variations can lead to various traits and health outcomes. While some can strengthen us, others can make us more vulnerable. We just looked at an example of a disease-causing variant, but not all genetic variants will cause disease. Some genetic variations can be beneficial, like a shortcut that helps us run faster or resist illnesses. For example, certain variations might make our immune system stronger, giving us an advantage against infections. These positive variations are like finding a special route in the maze that leads to better health or unique talents. Sometimes these variations are neither harmful nor beneficial. They're just neutral. They don't significantly change how we function. They don't provide an advantage or a disadvantage to our well-being. They're simply just part of what makes us unique. An example of an outcome from a neutral variant is hair color. While the variation in the gene determines whether someone has brown, blonde, or red hair, it does not impact our health or cause any disease. These neutral variations are like decorations along the maze that show off individuality, especially things like hair, height, or even how we smile. To us, these paths are ordinary. Harmful variants or mutations are genetic changes that may lead to vulnerabilities such as health issues or genetic disorders. For example, a harmful variant can occur in a gene responsible for digesting food, such as the lactase gene, which helps break down lactose. A mutation in this gene can cause lactose intolerance, where the body can no longer properly digest lactose, leading to digestive issues like bloating and discomfort. This is much like taking a wrong turn in the maze that leads to an unexpected obstacle and prevents you from continuing smoothly. In summary, mutation refers to a permanent DNA change, often linked to disease, while variant is a broader term for any genetic difference, which can be beneficial, neutral, or harmful, shaping our traits and health outcomes in various ways, like taking different paths in the maze. The science of genetics is still fairly new, and we still have so much to learn. Because of this, we don't know every single genetic change that exists. Sometimes we encounter variants that are still a mystery, known as variants of uncertain significance, or VUS. These genetic changes haven't been fully studied yet, and scientists are still working to understand their impact. They could be harmless, helpful, or harmful, but for now, the outcome is unclear. It's like coming upon an unknown path in the maze. We don't know where it leads yet, but with more exploration, we can map the way and uncover its true direction. The research scientists are doing is like that ongoing exploration, gradually creating a clearer map of what each path might mean. In this ever-changing genetic maze, understanding our pathways, or our genes, helps us navigate life more effectively. With knowledge of these routes, we can better understand ourselves and the potential impact of genetic variations on our health, whether they lead to strengths, challenges, or just what make us who we are. Now that we've gone over the types of genetic changes, let's look into where these genetic changes can happen. Let's explore two main types of genetic material in our maze, germline and somatic. Think of these two as different entrances at the start of our journey. The first entrance is the germline entrance. A germline mutation is a change in the DNA that occurs in a reproductive cell, which is the sperm or the egg cell. These can be passed down to our offspring. These mutations can affect multiple generations, influencing inherited traits and health conditions, such as eye color or genetic disorders. This is like finding a special path in the maze that can be shared with others. If a parent takes this path, they can pass it down to their children. Now let's look at the second entrance, somatic. A somatic mutation is a change in DNA that occurs in non-reproductive cells, like skin cells or muscle cells. These are not passed on to offspring. These mutations are confined to the individual and do not affect their descendants. This is like taking a path in the maze where any changes you make only affect your journey, not those of others. For example, if you fall and get a scratch or bruise in the maze, it doesn't impact anyone else or get passed down to future generations. Now, let's look at where this genetic information is stored. Our genes can exist in different areas of our cells, and genetic changes in each of these areas can have different effects. There are two main places in our maze: nuclear DNA and mitochondrial DNA. Nuclear DNA is the genetic material found in the nucleus of a cell, and it contains the instructions that direct the cell's activities and functions. It holds the majority of our genetic information, determining traits and guiding processes like growth and reproduction. In us, nuclear DNA is like a central tower that holds all the maps and blueprints, directing the flow of the entire maze and is in charge of how each part operates. Mitochondrial DNA is the genetic material found in the mitochondria. These are the energy-producing structures within our cells. Unlike nuclear DNA, mitochondrial DNA is inherited only from the mother and is responsible for producing the energy that cells need to function. Mitochondrial DNA is like the small power stations along the maze, providing energy to keep everyone and everything running smoothly. So whether it's through germline or somatic paths, and whether the information is stored, all types of genetic material influence our journey through life in different ways. Now that we know where genetic changes can happen, you may be thinking, what exactly are these changes? How are they affecting our genetic material? We have discussed that mutations can change our course through the maze. Some are neutral, some are helpful, while others can be harmful. Each mutation can affect our health differently, and we categorize them into several types. For the sake of simplifying this complicated information, we have placed them into three broad categories. Changes to the chromosome can involve large alterations in the structure or arrangement of chromosomes, known as chromosomal changes. Copy number variants refer to the changes in the amount of genetic material within a gene, while point mutations are small changes to the DNA sequence, affecting the specific spelling of a gene. These variations can lead to different traits or health conditions depending on the nature and location of the change. Let's explore different types of chromosomal changes that can impact an individual's genetic makeup. These include aneuploidy, where there's an extra or missing chromosome, translocations, where a piece of one chromosome is swapped with a piece of another, and inversions, where a section of a chromosome is flipped upside down. Each of these changes can have significant effects on an individual's traits or health. Aneuploidy refers to an abnormal number of chromosomes in a cell, either an extra chromosome or a missing one, which can affect development and health. Normally, humans have 46 chromosomes arranged into 23 pairs, one pair from our mother and one pair from our father. But with an aneuploidy, the total number is altered. And this can lead to conditions like Klinefelter syndrome seen here. This is a condition where males have an extra X chromosome, XXY instead of XY. These changes can result in a wide range of physical, developmental, and cognitive effects depending on which chromosome is involved. Aneuploidy is like navigating the maze with an extra or missing path, disrupting the journey and leading to unexpected challenges or detours along the way. Translocations occur when a piece of one chromosome is swapped with a piece of another, changing the usual structure and arrangement of the genetic material. Depending on the type of the size of translocation, it can disrupt normal gene function or regulation, potentially leading to health issues. It's like swapping paths in a maze, which can alter how you reach your goal. As mentioned, there are different kinds of translocations, some of which may not change the overall outcome, while others can affect the layout or structure of the maze, but all still ultimately lead to the same destination. Inversions occur when a piece of a chromosome is flipped upside down, causing the genetic material to be rearranged. While the genetic information is not lost, the new arrangement can disrupt gene function or regulation, leading to potential health issues or developmental changes. It's like turning a part of the maze upside down. The paths are still there, but their order is reversed, making it harder to navigate. Now we will explore the types of copy number variants. Copy number variants, sometimes referred to as CNVs, refer to the changes in the number of copies of a particular gene or genomic region. We will cover deletions, where one or more genes are removed from the genome, duplications, where genes are copied or doubled, insertions, where an extra piece of DNA is inserted into the genetic material, and tandem repeats, where a small sequence of genetic material is repeated multiple times. Each of these changes can impact how the genetic code functions, potentially leading to various health effects or developmental changes, depending on the size of how crucial the affected genes are for normal function. Let's dive deeper into these. A deletion occurs when a segment of genetic material is lost or removed from the genetic material. This can involve one or more genes. Depending on the size of the deletion, it can disrupt important functions or processes. It's like a part of the path being erased, preventing you from following that route and making the journey more difficult or even impossible. Duplications occur when a gene or multiple genes are copied, resulting in extra copies of genetic material. This can happen when a section of DNA is repeated, either in a small or large scale. While this duplication can sometimes be beneficial by providing extra copies of important genes, it can also create confusion or disrupt normal function if the extra copies interfere with the proper genetic balance. This is like having a path that gets copied, creating a double route that leads to the same spot. Insertions occur when an extra piece of DNA is added into the genetic material, which can involve one or more genes. This extra segment of DNA can disrupt normal function by altering the gene sequence or introducing new genetic information, which can sometimes lead to beneficial changes, but may also cause problems if the inserted DNA interferes with critical processes. Insertions are considered copy number variants, but can also be considered a chromosomal change depending on the insertion size of new genetic material. It's like adding a new path to the maze that wasn't there before, potentially changing the route and adding complexity. Tandem repeats occur when a small sequence of DNA is repeated multiple times in a row within the genome. These repeats can range from just a few to hundreds of copies. While tandem repeats don't always change the meaning of the end goal, they can impact gene function or regulation. And in some cases, they can lead to diseases or developmental issues if the repeats become too long or unstable. In the maze analogy, it's like encountering paths that loop back on themselves and repeat, sometimes making the journey longer. Now let's look at different types of point mutations, which involve changes to the DNA sequence of a gene. These mutations can affect how the gene is read and interpreted by the cell. Missense mutations change the gene sequence, resulting in a different meaning or function. Nonsense mutations introduce a change that causes the gene to stop making sense, often leading to a non-functional protein. Silent mutations occur when the gene sequence is altered, but the change doesn't affect its function. We'll dive deeper into each one of these in the following slides. A missense mutation occurs when there's a change in the DNA sequence of a gene that causes it to have a different meaning, altering the protein structure and function. In the maze analogy, it's like taking a wrong turn that still leads somewhere, but the outcome is not what you expected. A nonsense mutation happens when a change in the DNA sequence causes the gene to stop making a protein too early, making a broken protein. This can prevent important proteins from being made properly and cause health problems. This is like hitting a wall that stops you from going any further, being unable to complete that portion of the maze. A silent mutation is a change in the DNA sequence of a gene that doesn't affect how the gene is interpreted or the protein it produces. Even though the gene sequence is altered, it still codes for the same protein, so it doesn't have any impact on the organism. It's like choosing a path that looks different but actually leads to the same destination. Nothing changes. Mutations in our DNA can have a wide range of effects on how our genes function. These changes, whether small alterations in the DNA sequence or large structural shifts, can lead to various outcomes. Some mutations result in a loss of function, where the gene or protein no longer performs its intended role, often leading to disease or dysfunction. Other mutations can cause a gain of function, where the gene or protein gains a new or enhanced ability, which can sometimes be harmful, but also provide an advantage in certain circumstances. So whether a mutation helps, harms, or changes our path a little, each one plays a role in our unique journey through the maze of life. Understanding these mutations helps us learn more about ourselves and our health. To find out more about the mutations in our maze, scientists use various genetic testing methods. Think of these tests as special tools that help us explore and understand the paths better. Just like you might use a map or a compass to navigate a complex maze, genetic tests help us look for specific changes in our DNA. Some tests can scan the entire maze for all types of mutations, while others focus on particular paths. If you're looking for more information on genetic testing methods and interpretation, please watch our other videos. Sometimes your genetic test results may come back as negative or unknown. Receiving results such as these does not mean that there is no answer. It may just mean we have not found the answer yet. You are an essential part of this journey. Regularly checking the status of these VOSs and engaging in discussions with healthcare professionals can lead to better understanding and management of your genetic health. So there are many common questions after receiving test results and/or a diagnosis. Many people wonder, where did this come from, or why did this happen? Understanding inheritance patterns is essential in answering these questions. When it comes to genetic changes, there are two primary ways that they can occur. Some changes just happen spontaneously, while others are hereditary, passed down from parents to their children. Think of it like navigating a maze. Sometimes you might stumble upon a new path that wasn't there before, and this represents a spontaneous genetic change, which can occur due to various factors like environmental influences, random errors in DNA replication, or even exposure to certain substances. These unexpected changes can lead to new traits or conditions that may not have been present in the family before. On the other hand, some paths in the maze are well trodden, having been used by many generations. These represent hereditary genetic changes. If a genetic condition is hereditary, it means it can be passed down from one generation to the next, often following specific inheritance patterns. For example, if a parent has a hereditary condition, there's a chance that their children may inherit it as well. It's also important to note that all conditions are genetic, which means they involve our genes in some way. However, not all genetic conditions are inherited. Some conditions do arise in a spontaneous de novo fashion. This distinction can help clarify why certain conditions appear in an individual, even if there's no family history of the issue. We've talked about where, what, and how genetic changes occur and become, and how our environment plays a role in our health. This is another layer of complexity to consider: multifactorial mutations. These are not caused by a single genetic change, but rather by a combination of genetic and environmental factors working together. Multifactorial mutations are genetic changes that result from a combination of multiple factors, including both genetic and environmental influences. Unlike single gene mutations, which are caused by changes in a specific gene, multifactorial mutations involve interactions between various genes and environmental factors such as lifestyle, diet, and exposure to toxins. These types of mutations can contribute to complex traits and conditions like heart disease, diabetes, and many common disorders where multiple genes and external factors work together to affect the outcome. Essentially, they highlight how both our genes and our environment play a role in our health and traits. Imagine multifactorial mutations as different tools you can use at various points along your journey through the maze. Each tool represents a factor that can affect your progress, like a flashlight for dark areas, a map for guidance, or a ladder to help you overcome obstacles. As you navigate the maze, choosing the right tool at the right time can change how you move through it. For instance, using a flashlight might reveal hidden paths, while a ladder can help you overcome barriers. However, some tools may not always be available, and certain environmental factors that influence your journey, like weather conditions or unexpected obstacles, aren't always within your control. Epigenetic changes are modifications that affect how our genes are expressed without changing the actual DNA sequence. These changes can be influenced by various factors, such as our environment, lifestyle, and experiences, and can have a significant impact on our traits and health, even though the underlying DNA structure remains the same. Imagine the maze as a series of paths that can be marked with signs indicating which routes to take or avoid. These signs represent epigenetic changes. In our maze, some paths may be marked as open or closed based on weather or construction. Similarly, epigenetic changes can turn genes on or off, which just determines whether a particular gene is active or inactive. Thank you for joining us in this exploration of genetic materials. We understand that the world of genetics can be complex and difficult to navigate, with many terms, concepts, and implications that can feel overwhelming. But remember, learning is a journey, and it's okay to take your time to fully understand it. Keep learning, keep asking questions, and embrace the adventure ahead. Every step you take brings you closer to better understanding yourself and your health. As we navigate the intricate maze of genetics, remember that knowledge is power, whether it's through testing, research, or support. Take charge of your genetic journey. And for more information, we have resources listed here that can be invaluable for you seeking reliable and up-to-date information on genetic and rare disorders. For example, Medline Plus provides trusted health information, offering clear and accurate details about medical conditions, treatments, and genetic issues. Unique, Understanding Rare Chromosome and Gene Disorders offers specialized insights into rare genetic conditions, helping individuals and families better understand and manage these complex disorders. The National Organization for Rare Disorders, or NORD, offers comprehensive resources on rare conditions, supporting both patients and healthcare providers in navigating these challenges. And lastly, the Genetic and Rare Disease Information Center provides current, trustworthy information on rare diseases, ensuring individuals have access to critical health information for informed decision-making and care management. Exciting advancements are being made in gene therapy, aiming to correct genetic disorders at the source. While it's still a developing field, resources like clinicaltrials.gov can help you stay informed about ongoing studies. So if you're interested in learning more about genetics, inheritance, and testing methods, please also watch our following videos. Thank you so much and keep learning.